And now Kelly is going to talk about inverse modeling. So this is taking it even further from QTL mapping.
Okay, thanks, Mike. Just to wrap up, I would like to talk a little bit about inverse modeling. For those of you that don't quite understand what that is, I'm going to provide a basic introduction to it. But what we're essentially trying to do is extend the amount of useful information that we can use for the data analysis that Mike described. Our tractor is really, at the core of it, only collecting three pieces of information. We have the reflectance of radiation from the canopy, the temperature of the canopy, and the essentially distance from the canopy to a sensor, which is a surrogate for crop height. So how can we use our models, our physical and ecophysiological models, to estimate additional traits that can be used for phenotypic data analysis? And the answer may be using some kind of inverse modeling procedure. Okay, so here is a basic example of inverse modeling. Typically when a modeler is designing a model, the way that it's done is attributes of a particular system are identified. And those are the inputs to a model, and then the model is programmed to simulate some quantity or some value that can be observed about the system. So PROSAIL is an example model that's used quite often. It's a radiative transfer model used in remote sensing. This model basically takes as input things like the chlorophyll A and B content of a leaf, the water content of a leaf, the leaf area index of a canopy, and a few other parameters. And the model will then simulate what the canopy spectral reflectance response should be. And for typical vegetation, we get some response like this, where the wavelength here is 400 nanometers and out here is about 2,500 nanometers. So if we were going to do a forward modeling procedure, we would basically know all of these inputs. These various inputs about the canopy would be known. We would run the model forward and get the output. So very simple. But that's typically not the case, and it's not the case for phenotyping. For phenotyping, we're measuring reflectance. We're measuring what the model outputs. And so then what we need to do is use that information along with the model to estimate what the model input should be. So if a model can be mathematically inverted, then we can calculate that directly. However, most models used in the physical sciences are not able to be inverted because they are complex. And for that reason, we have to turn to optimization, which essentially means running this model forward many times until we find the solution or the input parameters that minimize the error between the observed and simulated model output. When we do optimization, that brings about a whole host of other questions that need to be addressed, such as which model parameters do you adjust, which ones do you constrain, for the ones that you adjust, what bounds do you allow them to be flexible within. For the case of PROSAIL, what spectral wavelengths do you include in the optimization, when do you observe the canopy, at what time, and what objective function are we minimizing or maximizing. Depending on the decision for all of these questions, the outcome of the optimization will be affected, and so we need to very carefully address each one of these. So my basic methodology to test this approach has been to use the data that we have collected in the field. Again, this is for our cotton experiments. We have 3 years of cultivar by water experiments. And I have gone out to the field and collected hyperspectral reflectance information of the crop canopy. This is actually separate from the tractor because this instrument is much more expensive, and we can only afford to have it carried in the field. So I've carried this instrument in the field and collected spectral reflectance responses of the canopy. I've then taken that data back into the computer lab and built a computational program that implements PROSAIL. It implements an optimization algorithm called PEST and uses Python for basic control to conduct these inverse modeling or optimization experiments. So the basic procedure has been to first evaluate those questions that I asked in the previous slide and try to figure out what is the best protocol for the optimization. Then we can do things like compare measured and simulated results to make sure the model is performing adequately. And importantly, we can use statistical tests to test the significance of optimized model parameters, which essentially are other traits that we're observing through this exercise of our canopy. So here again is a little bit of a summary of our field experiment. We have 3 years of data from 2010 to 2012, well-watered and water-limited irrigation on cotton and many different cotton cultivars. I'm going to be focusing here on observations collected in the AM and PM on August 4th and in the AM on August 18th, 2011, 2 different years. The instrument that I'm using is an ASD Field Spec Pro, and it essentially measures reflectance of light between 350 and 2500 nanometers at a 1 nanometer bandwidth. This is a picture of us using a spectral on panel to correct that data against the incoming solar irradiance at the time of measurement. We also have some other data to go along with our spectral measurements, including leaf water content, specific leaf area, and chlorophyll A and B content. A little more about the PROSAIL model. PROSAIL is a combination of prospect, which is a model of the way that light interacts within a plant leaf, how the plant leaf structure treats light, reflects it, absorbs it, and transmits it. And the SAIL model, which is essentially a model of how solar angle, solar geometry affects how light is reflected from a plant canopy. 14 input parameters are required that describe leaf pigment, leaf water content, canopy architecture parameters, solar geometry parameters, and some others. And then as an output from this model, we get the canopy bidirectional reflectance from 400 to 2500 nanometers in 1 nanometer increments. So the output of this model corresponds very nicely with the observations that we're collecting with our hyperspectral equipment. So to parameterize the model, I'm holding several parameters fixed. Some other parameters are based on observations in the field. And the key thing to notice is that I'm going to be optimizing 3 parameters that are basically an N parameter, which is a structural parameter of the canopy. LAI, leaf area index, and the leaf inclination angle will be optimized to minimize error between our observed spectral data and our simulated data. So this diagram kind of shows the procedure that we're doing. We implement our PEST optimization routine. Into that goes the output spectral response from PROSAIL and the observed spectral response from our spectroradiometer in the field. We then use the optimization to intelligently adjust these 3 parameters that we're optimizing. And then this happens many, many times until we come to a solution for the optimization where we've adjusted these 3 parameters to minimize the error between the measured and simulated canopy spectral reflectance. And we're going to be focusing here on 3 different groups of bands, 400 to 1300 nanometers, 1450 to 1770, getting into the shortwave infrared. 400 to 1300 is more in the visible NIR, and 1970 to 2400 nanometers is on out into the further into the shortwave infrared. Then what we can do is some very routine ANOVA and multiple comparisons tests on our resulting trait estimates that we're getting through our model optimization. Here I'm showing statistically significant differences between LAI, between our well-watered and water-limited conditions for the August 4th date at 2010 a.m. And we have similarly some statistical differences among 4 cultivars. That's important because essentially that's what we're trying to do here, is find statistical differences among the cultivars, and that information, the traits then that we're estimating, can be used in further genetic analysis that Mike described. One of the issues, if I go back, I want you to notice, is that the LAI in the morning is about 4 to 5. In the afternoon, on the same day, we're getting an LAI, optimized LAI of 6 or more. Of course, that is not realistic because we don't expect the canopy to grow that much in an afternoon. So this highlights one issue with optimization, is that you have to be very careful about the values that you're getting, making sure that they are realistic. Again, changing the protocol, the procedure for the optimization can highly affect what values we actually get. The important thing to notice is that we're getting statistically significant differences from these trait estimates among our treatments in the field. For the PM, we actually have now statistically significant differences between our leaf angle, probably due to the increased leaf droop due to water stress in the afternoon. And on into the second year, we're again seeing more statistically significant differences. Okay, so in conclusion, we can maybe use inverse modeling to find statistically significant differences between trait estimates that are more difficult to observe in the field. And that's where the modeling comes into play. However, the results are highly dependent on how you set up the optimization routine. Plant growth models were mentioned earlier. I think plant growth models could also help in this case. If we know the temporal time series of a particular, say, LAI, a state variable in a model, that might help constrain the way that PROSAIL is estimating LAI through inverse modeling. So with that, I'll conclude and turn it over to Sean. Thank you.